So you may wonder, why is this happening? Okay, why is it W shape? Well, here's the reason. Okay. So when the cutoff is 0, then you basically have no choice. Why? Because here's why. So here's the fence inequality, right? If C is 0, the only choice you can have, every time you have to choose the baseline model, because you want this thing to be exactly 0. Okay, so because nobody can do better than the baseline model, but there is a model that does exactly as well, and that's the baseline model itself. Okay, so when the C equal to 0, every time you make a model selection, you always choose the baseline model. So that probability is 100%. Okay, so that's why the empirical probability is 100%. Okay, you don't have a choice. Okay, and also when C is very large, once again, you don't have a choice. Why? Because, for example, let's just consider this quite minimum dimension criterion. Okay, if C is very large, then what happens is that every model is in the fence, because C is a very large number. Everybody satisfies this inequality, so everybody's in the fence, right? So then, how do you select a model how do you select among the models within the fence? Well, you say we choose the model that is the simplest. Okay, so then here we go. So every time you choose the simplest model, regardless how the model fits the data, because you always choose the same model, which is the one with the minimum dimension. So again, you don't have a choice. Every time you choose the model with the minimum dimension, that is why the empirical probability is equal to 1. Okay, so basically this illustrates how to choose the cutoff adaptively. Now, one critical assumption of this method that was originally proposed by this paper, published in the Annals of Statistics, is that there exists a true model among the candidates. But this assumption, although it's necessary in establishing consistency of the model selection, it limits the scope of applications, because in many cases, the true model may simply not exist, or exist but not among the candidates. How could you know that the true model is among the candidates? In many cases, this is not practical, right? So the question is, can we use the fence method without this assumption, okay, without requiring the existence of a true model among the candidates? And this would be the case, in particular, for the problem of microarray gene set analysis discussed in the sequel. Right. So now let me introduce a motivating example. Here's a main motivation for our gene set identification problem. So it's a data set involving a data set used in a study in tracking pathway involvement in late stage versus early stage colon cancers. Basically, we have microarray gene set expression data from 4 distinct colon tissue samples. So these are the 4 different colon tissue samples. So these were collected. So then the actual data set consists of 104 samples, of which 25 were B survivors, 21 were Duke Cs, 35 were Duke D, and 23 were liver mets. And these were all collected at the Ireland Cancer Center of Case Western Reserve University, where Dr. Rao used to collaborate with some scientists over there. There were a total of 59,618 probe sets or genes. So these are so this is a brief description of the data. So then one question of particular interest to colon cancer biologists is to see which colon cancer specific pathways. Here by pathways we mean groups of genes acting in concert with one another seem to be at play when comparing a bad prognosis tumor to a relatively good one. And understanding which pathways are differentially expressed gives a better picture of the biological processes that might be at play in worsening prognosis. So basically, this is the motivating example, which I'll come back at the end if I have time. So now, once again, as I mentioned, in order to apply our method, more specifically the FANS method to microarray gene set analysis, we need to extend the FANS method to the situation where a true model may not exist. Okay? The extension can be done, but it involves a little bit technical treatments or mathematical definitions. So what I decided to tell you briefly the idea of the extension, then I'll skip the details. Okay? So basically, it's all about how you define the term true models. Okay? So if you define the true model in terms of the traditional sense, traditional definition, so then you run into this trouble of having to worry about the existence of true model. Okay? So now, if you define a true model in terms of the model that best fits your data, then such a model always exists, right? So in a way, that is not necessarily a true model in the traditional sense, but the one that can be best approximated. Okay, so this is the definition. Okay, so you have a group of candidate models, okay, among which there is a one that best approximate your data. Okay, so this is what we call the true model. Okay, but this one may not be the most efficient one. Okay, so that's why we need to do a model selection. Okay, so this way you extend the notion of model selection or true models, and therefore avoids the requirement for the existence of true model, because such a true model always exists. Okay. So some technical terms which I'll skip. Right. So now let me go back to the microarray gene set analysis. There have been studies on the problem of identifying differentially expressed groups of genes, which we call gene sets, from a set of microarray experiments. One of the papers is this one, which I discussed with one of the faculty members earlier today. So basically, these problems are fairly practical from many of the recent studies. In particular, there's this paper by Efron and Tipshrani, published in 2007, in which they propose a method called gene set analysis, or GSA, based on testing the significance of gene sets. Now, the method proposed by this paper is called GSEA, gene set gene expression enrichment analysis. Well, this one is called gene set analysis. So there's a one letter difference, but the methods are different. And Efron and Tipshrani claim that their method performed better than GSEA based on their studies. Now here's the problem formulated in a mathematical formation. Suppose there are n genes, capital N genes, measures on small n microarrays under two different experimental conditions called control and treatment. The number of genes is usually large, say at least a few thousand, while this little n, which corresponds to the sample size, is much smaller, say 100 or fewer. Now, the interest is to assess the significance of some predefined gene sets rather than individual genes in terms of response to the treatment. The gene sets are derived from different sources, such as biological pathways. Here's the description of the GSA procedure. So first you compute a summary statistic for each gene. For example, the two-sample T statistics. So then, based on the gene set, based on the gene-wise statistics, you come up with a gene set statistic for each gene set. That is computed for each gene set based on the summary statistics. For example, you can take the average of all the summary statistics within that gene set. Okay, this gives you a gene set statistic. The next step is called restandardization. You do this by subtracting the gene-wise mean of the summary statistics from each gene set statistic, and then dividing the difference by the gene-wise standard deviation of the summary statistics. Results of the restandardized gene set statistics are called gene set scores. The p-value for each gene set score and the force discovery rate, or FDR, applied to those p-values are then estimated by permutations of the class labels in the control and the treatment groups. Depending on the FDR, a number of gene sets may be declared significant, or no gene set is declared significant. So this is a description of the GSA method. So now, so our goal is to take another look of the FANS method, and this will allow us to be able to use our method for the current problem of gene set identification. Now here's the another way to look at the FANS method. So basically, you ask yourself the question of how to choose a model within the FANS. Okay, I'm going to make it a little bit simpler. Let's just say, remember there's a criterion of optimality to choose the model within the FANS. Okay, so let's just say the criterion is the minimum dimension.